Okay, so I will probably we will follow the program that I've sent you. So I will start with a brief introduction of ARISTO, and then the floor will be with four PhD students that they will give you a brief results, briefly some of the highlights of their research, and then we can have time, I would suggest, for discussion and exchange of ideas. So do you see my screen? Yes?
Yes.
Yep.
Yes.
Yes.
Excellent. Okay. Okay. Right. Okay. So so the intention of my presentation will be actually to give you a brief introduction of ARISTO, what actually triggered the formation and the submission of this project, and very briefly what the different fellows, the different PhD students in ARISTO are dealing with. So so we all know that at the moment at the regulatory level, we determine if a pesticide creates undesirable effects on the soil microbiota using the N-mineralization test, which dates back to the 50s or 60s, if I'm right. And it actually provides a lumped measurement of the transformation of organic nitrogen to nitrate. So it's a lumped measurement of ammonification and nitrification process in soil. So I'm not going to go through the details of what is the N-mineralization test. I think we all know every detail of it. However, in light of the major advances in soil microbial ecology and soil microbiology in the last 20 years, EFSA identified that there should be an update on the regulatory framework of the assessment of pesticides toxicity on soil biota, including soil microorganisms. And the major conclusions of this EFSA document was that the N-mineralization test is inclusive and will be maintained. New molecular methods and biochemical tools are available, but not yet standardized. And these first two conclusions, in my view, they are pretty much outdated and not and incorrect. And then they suggested arbuscular mycorrhizal fungi as potential functional bioindicator group for assessing the soil microbial toxicity of pesticides. And in ARISTO, we argue that this is probably true, but we need some. We can do some more with microbial indicators. And finally, they suggested that functional endpoints are more easily quantifiable than by diversity data, which is really true. So basically, we thought when we were writing the project, what really do we need to assess the soil microbial toxicity of pesticides? So the key elements of a good recipe is standardized methods and tools to do that, microbial indicators that could serve as indicators for assessing the toxicity, because we cannot test everything, and a risk assessment sim for decision making. So regarding standardized tools, we already just list here the different ISO standardized methods that they are available, and you can see some molecular tools that they are available for inclusion in risk assessment, and some other tools that they have been there for quite a while now. Then the second element I mentioned is bioindicators. Of course, EFSA proposed arbuscular mycorrhizal fungi. They seem to be responsive to abiotic stressors. They are obligate plant symbiotes. There is an ISO standardized methods, the spore germination test for testing toxicity of pesticides or other pollutants. and they are very important parts and fraction of the soil microbiota, especially in low-input agricultural systems. In addition to IMF, in ARISTO, we posit that another very promising group of microbial indicators is ammonia oxidizers. They are they have been proposed from several different groups, including us in the past. They are composed of three different subgroups: ammonia oxidizing bacteria, ammonia oxidizing archaea, and recently we know that there are Comamox bacteria that they contribute also to ammonia transformation. And they have very good credentials as bioindicators, since they mediate the rate-limiting step of nitrification. They are sensitive to biotic and abiotic perturbation. This is something that we know for quite a while. I came across a paper from a group in US back in the 70s that they are indicating this particular sensitivity of nitrosomonas in organic pollutants, including pesticides. And in fact, we have very good standardized tools for the measurement of their activity and their abundance biomass in soil. So the third element I mentioned was a tiered risk assessment approach, and our group, before actually the start of ARISTO, has proposed a tiered risk assessment approach where we have a tier 1 in vitro testing. Then at tier 2, we go at lab soil testing. At tier 3, if needed, we go at field testing. So tier 1, it's in vitro assessment with single organisms, like single species tests. And then you can increase complexity by moving to soil experiments in the lab and in the field with increasing complexity. And in all these tiers, we always testing is focused on arbuscular mycorrhizal fungi and ammonia oxidizing microbes as key microbial indicators. So this was actually the core of ARISTO project. The core work plan or ARISTO project was built around this tiered risk assessment approach. So you can see here that we start at work package 4 with in vitro testing. Then we move to single microbial group testing in soil scale. And then further, we increase the complexity and we move at microbial networks and soil food web level, looking at effects at ecosystem scale. And in ARISTO, we also address some, I would say, very much emerging areas of pesticide research, which was the soil microbial toxicity of pesticide mixtures or of biopesticides, but also assessing the soil microbial toxicity of transformation products. So that was the core work plan of ARISTO. And where you see ESR, it stands for early stage researchers, or the PhD students that you will hear after me. So the major goal of ARISTO was to produce all the benchmarking knowledge that we need and to develop advanced tools to assess the toxicity of pesticides on soil microorganisms. And as you realize by the presentation or when the students presented themselves, you realize that the students are sharing their time between industry and academia. So we have 9 PhD students. The project is in the 4th year now, and we've got 7 academic partners and 9 industrial partners. And the students, each of the 9 students had to spend 18 months in an academic partner and 18 months in an industrial partner. In between that, they were allowed and they were actually encouraged to spend time in some of the associated partners that we have in the project. These partners, they don't claim part of the budget, but they offer secondments to the different students in training in different skills that they might be useful for their PhD. So I will very quickly go through the different students that we and present very quickly what they are doing, and then I will give the floor to the 4 students that they will present. Unfortunately, we don't have time for all 4 or 9 students to present, so we selected some of them that they will give you insights in what we have been working on in the last 3 years. So at tier 1, we have Eleftheria and Marjan that you will both hear that they are working on the standardization and the implementation of tier 1 testing on ammonia oxidizers and arbuscular mycorrhizal fungi. At the second the work package 5, we have Laura and Anna that they actually perform experiments to assess the toxicity of pesticides on soil microbes and against natural soil microbial assemblages. And Laura is working with ammonia oxidizers and Anna with arbuscular mycorrhizal fungi. And then we move to ecosystem level assessment of potential effects of pesticides, where we have Kara that is assessing effects within microbial networks, and Marta that is looking at effects on interacting predator-prey systems like protist bacteria systems that they are very interesting to look at in soils. Then we have Camila that is working on the effects of pesticides of pesticide mixtures on the soil microbiota, and Alex that is working on to assess the impact of biopesticides on the soil microbiota, and particularly of botanical compounds. And finally, Kuni Yang brings in expertise on in silico, the development of in silico tools for assessing the soil microbial toxicity of pesticide transformation products with a focus, but he will explain you more. He will give you more insights with a focus on ammonia oxidizing microbes. So what are you going to hear today? You will hear from Marjan and Eleftheria about the development of tier 1 tests for ammonia oxidizing microorganisms and arbuscular mycorrhizal fungi. Then you will hear from Kara about direct and indirect effects of pesticides on the soil microbiota, and eventually we have Kuni Yang that will present you the development of a tool for in silico prediction of the toxicity of pesticides and of their transformation products on the soil microorganisms, with a main focus on ammonia oxidizers. So eventually, the expected outcome from ARISTO would be to implement a new tiered risk assessment sim with increasing level of complexity, supported by scientific evidence produced by ARISTO. Implementation of and use of microbial indicators of arbuscular mycorrhizal fungi and ammonia oxidizers as proxies for assessing the toxicity of pesticides to the soil microbiota. And of course, the introduction of the standardized methods and tools that we developed in ARISTO and made them available in a future revision of the risk assessment sim for soil microbes. So that's all from my side. I would suggest that we move to the presentations of the students, and then at the end we will have a half an hour or so for discussion. Is that okay for you? Okay. So maybe the next presentation could be done by Eleftheria and then Marjan. So Eleftheria, do you want to start?
Yes. Do you see my screen? So, well, today I will present you a part of my PhD with title In Vitro Assessment of Pesticide Toxicity on Non-Target Defining Microorganisms, a Novel Tool for Ecotoxicity Risk Assessment. I will start with some theoretical information Pesticides are a significant part of modern agriculture and are heavily used in agro-ecosystem to ensure high yield and meet growing global food demands. However, only 5% of these applied compounds reach their target, with the remaining ending up in soil with possible negative effects. After application in soil, pesticides can interact with soil microorganisms with fundamental importance for environmental pollution and ecosystem. On one hand, soil microorganisms can degrade pesticides and use them as energy source for their growth. And on the other hand, pesticides can negatively affect soil microorganisms by inhibiting their activities like nitrification. And also, this compound can change the soil microbial community structure with serious consequences for environmental functioning. So pesticides are considered major environmental pollutants, and their toxicity is regulated by European Union framework, which is well established for aquatic and terrestrial organisms. However, there are no corresponding bioassays for soil microorganisms, mainly due to the lack of identified soil microbial indicators, even though that soil microorganisms were identified as a specific protection goal for environmental risk assessment by EFSA in 2010. In 2016, EFSA released a first draft in which a vascular mycorrhizal fungi can be used as an indicator of pesticide toxicity using advanced molecular tools. And at the same time, some studies propose that the monoxidizing microorganisms could be ideal candidates for agrochemical toxicity. The output of this study proposed a new risk assessment scheme in which in vitro tests using the most sensitive and representative ammonia-oxidizing microbes and the vascular mycorrhizal fungi could be the first testing step before proceed in soil experiments for the toxicity of pesticide, both at lab and field scale. Nitrification is the key process of nitrogen cycle and was considered a two-step process. The first step involved the oxidation of ammonium to hydroxylamine via ammonium monoxygenase, and then the transformation of hydroxylamine to nitric oxide and finally to nitrite. This step is carried out by ammonia-oxidizing bacteria, AOB, and ammonia-oxidizing archaea, AOA, and is the rate-limiting step of nitrification. The second step involves the oxidation of nitrite to nitrate by nitrite-oxidizing bacteria, NOB. In 2015, another microbial group was reported that participates in the nitrification process. This group is called COMAMOX, complete ammonia oxidizer, and has a metabolic capacity to perform both steps of nitrification in one cell. But why ammonia oxidizer are considered ideal microbial indicator for pesticide toxicity? First, as I mentioned, they participate in the rate-limiting step of nitrogen cycle. We have good knowledge of their life cycle, ecology, biochemistry, and physiology. They show sensitivity to xenobiotic compounds, and we have well-standardized tools to measure their activity and abundance, both at in vitro and in soil level. So, in this study, in collaboration with the University of Thessaly and Professor Karpouza's lab, we tried to establish and standardize single-species in vitro tests using ammonia-oxidizing bacteria archaea and nitrite-oxidizing bacteria as a first-tier tool for assessing the toxicity of pesticide and soil microorganisms. We hypothesized that at least one strain within and between the different microbial group of identifiers will be systematically more sensitive and exhibit a clear ecological response to pesticide exposure. So for this purpose, we monitor the activity of 12 nitrifying strains growing in liquid culture via nitrite production in case of AOB and AOA group, or consumption in case of NOB group, over a broad range of pesticide concentration. Specifically, we used 6 AOB strains, 3 AOA, and 3 NOB strains, representing the dominant linkages found in soil ecosystems. Based on the nitrite measurements, we prepare the growth curves of the different strains in presence on the pesticide. We calculate the EC50 value, the concentration of the pesticide that reduce half of the activity, and we identify the most sensitive strain per group. In this table are presenting the pesticide that we choose for these experiments. The choice of pesticide was based on their mode of action, which seems to be relevant with ammonia oxidizer, and also these compounds are widely used in agricultural practice in Europe. The concentration level was calculated based on the recommended dose used in agricultural practice, and we test 4 concentration levels, 0.1, 1, 10, and 100 times of the recommended dose. It's important to say that for some compounds, we had some solubility problems, so we adjust the maximum concentration. We also accepted the activity of the strains. We also studied the degradation of the compounds in liquid culture via HPLC. I won't show you the results, but generally we observed that the compounds were stable in the liquid culture, with only exception pyraclostrobin, etradiazole, and mexazole, which they degraded in nitrobacter dark culture. Then, based on the EC50 values, we observed different toxicity patterns of pesticide categories to the defining groups. Insecticide showed higher toxicity against the AOA group compared to AOB, while fungicide showed high toxicity to both AOA and AOB group. In case of herbicide, we didn't observe significant difference in the toxicity to the different defining groups. Then we observed different sensitivity for different defining groups for the pesticide category. AOA showed higher sensitivity to insecticide and fungicide. AOB showed higher sensitivity fungicide, while NOB showed higher sensitivity to insecticide compared to herbicide. Then, again, based on the EC50 values, which are presented on these heat maps, we identified the most sensitive strain per group of nutrifiers. In case of the AOA group, we observed that the acidophilic Nitrosotala sinensis was the most sensitive, with the lowest calculated EC50 values for almost all tested pesticides. Neutrophilic AOAs, Nitrosocosmitis franklandianus and Nitrosophiera vienensis, showed the same toxicity patterns in all tested pesticides, with only exception for mexazole, which was more toxic for Nitrosophiera vienensis. Etradiazole, chlorpyrifos, and pyraclostrobin were the most toxic compounds to all tested AOA strains, with their EC50 values being significantly lower than the recommended dose of the calculated and used for the strains. In case of the AOB group, we observed that the sensitivity was pesticide and strain dependent, with Nitrosomonas aureia and Nitrosospira briensis being the most sensitive. Nitrosospira is often the dominant AOB link in the ecosystem. Therefore, Nitrosospira briensis seems the ideal candidate for risk assessment of pesticide and soil microorganisms. For compounds, methylfluoromethyl, 5-TCA, pyraclostrobin, and etradiazole were the most toxic compounds against all tested AOB strains, with the EC50 values of pyraclostrobin and etradiazole being lower or equivalent to the recommended dose. In case of NOB group, we observed that the effect of pesticide were weaker compared to AOB and to AOA. Nitrobacter SP and HB1 strain was the most sensitive to all tested pesticides, except glyphosate, AMBA, 3,5-DCA, which were more toxic for Nitrospira diffluviv. So, to sum up, the tested pesticides affect at least one non-target ammonia-oxidizing microbe, with AOA and AOB strains showing various levels of sensitivity because of their structural and biochemical differences. 3 strains, Nitrosotala sinensis from AOA group, Nitrosospira briensis from AOB group, and Nitrobacter sp and HB1 from NOB group were selected as bioindicator due to their high sensitivity, and they can use in our proposed single species test as a standard tier 1 ecotoxicity assay for providing a conservative assessment of potential toxicity of pesticide and soil microbiota. Our proposed single species test involves the exposure of the 3 strains in a different level of pesticide concentration and monitoring the activity of the strains in presence of pesticide by nitrite measurement, and also monitor the stability of the pesticides on liquid culture. Regarding the ongoing work of my PhD, we are assessing the impact of additional pesticide on the most sensitive strain as a proof of concept of our single species test. Here on this table are presented the pesticide that we have chosen for this and the concentration level. We are also investigating the toxicity mechanism of some compounds on the most sensitive AOB and AOA strains using transcriptomic and proteomic in order to see the regulatory response and the metabolism of the strains in presence of the pesticide. We have chosen chlorpyrifos because it showed high toxicity to all tested AOA strains, and methylfluoromethyl and pyraclostrobin because it showed high toxicity to all tested AOA and AOB strains. Finally, we'll compare our results with the effect of pesticides on ammonia oxidizer strain with a commercial available toxicity kit MARA, which is provided by NCIMB. I would like to thank all the people who involved in this project and, of course, you for your attention.
Thank you, Eleftheria. Okay, so keep notes and questions, and we will get back to that when we are done with the presentations. So I will give the floor to Marjan. Well, she will talk us through the in vitro testing against arbuscular mycorrhizal fungi.
And thank you, Dimitrios. I'm going to present you the novel tools for ecotoxicity risk assessment that we have used in the project in vitro assessment of the toxicity of pesticides on arbuscular mycorrhizal fungi. First, I'm going to start with a little talk about arbuscular mycorrhizal fungi. As Dimitrios also presented for us, it's an applicate biotrophic fungi that make symbiotic association with the vast majority of agricultural and horticultural crops. It's from the phylum Glomeromycota. So when the spore is in the soil, it can germinate and produce hypha in the presence of roots of plants. This hypha can penetrate inside the root cells and produce a specific structure called arbuscules. With this arbuscule, they can make this symbiotic association with the plants. During a time, and they can produce mass of extraradical mycelium inside the soil and intracellular mycelium inside the roots. Why they are important for us? Because they can make the absorption of nutrients from the soil to the plant 300 times more than the time that the plants absorb them themselves. Also, they can help plants in controlling the biotic stresses like pathogens and abiotic stresses like water stress, pollution, and salinity. On the other hand, using pesticides are inevitable in agriculture for having more yields and also to control the pests and pathogens of plants. So it's important for us to see if these pesticides can affect arbuscular mycorrhizal fungi as a bioindicator in a positive, negative, or neutral way. So to achieve this, we have implemented some experiments. One of them is to verify the impact of 19 pesticides containing fungicides, herbicides, and insecticides on the spore germination, which is the first level of the life cycle of AMF, specifically on Rhizophagous intraradices. We have used controlled water because aminomethylphosphonic acid and glyphosate were soluble in water, and also acetone because the rest of them, they got dissolved in acetone. So when we dissolved them in 5 different concentrations, we added them in MSR medium, and we solidified them inside the 12 wells microplates. We had 3 replicates of them. So we separated the spores of AMF from the 4-month-old bicompartment petries of intraradices, and we cut them in single spores and we transferred them to each well of microplates. Then we tracked their germination from the first day to 28 days. So based on the results, mostly herbicides and insecticides, they had no inhibiting effect on Rhizophagous intraradices. Like hymexazole is a fungicide that had this effect. We had also some delay effects like etridiazole that in 10 times and 20 times more than the field concentration, they started germination after 2 weeks. Some of them had a strong inhibiting effect at the higher concentrations, like iprodion, that in 10 times and 20 times more than the field concentration, they stop the germination. And finally, phyllodioxonil as a fungicide, it had a strong inhibiting effect even at the lowest concentration, that in all 5 tested concentration, the rate of germination was 0. To complete these results, we made the EC50 value table. As I told you and explained, herbicides and insecticides, they were almost with no inhibiting effect. And that's here, their EC50 value is higher than the highest dose that we have tested, so it means they are less or non-toxic. But on the contrary, for the fungicide, the fludioxonil is the one that I said in all 5 concentration, the rate of germination was 0. The EC50 value is lower than the lowest dose that we have tested, so it shows that fludioxonil is highly toxic for Rhizophagus interradices strain that we have tested. Then based off the other fungicide that we have tested, we implemented another experiment to verify the impact of fungicide on phosphorus uptake by extra-radical mycelium in vitro conditions. So we planted Medicago truncatula seeds in MSR medium in vitro, and after 7 days, they made plantlets. So we transferred the plantlets inside the bi-compartment Petri plates with a root compartment and hyphal compartment that they have a barrier in between. So we place the roots of the plant in the root compartment on the inoculum of Rhizophagus interradices, and the upper part of the plant was kept outside a sterilized situation. After 2 months, the roots of plants got colonized by AMF, and the hypha of AMF, they started to pass the barrier and go to the hyphal compartment. In this stage, we added 12 milliliter liquid medium every week to help them to grow in the hyphal compartment. After 4 weeks, when we had a mass of mycelium in the hyphal compartment, we removed the liquid medium and we washed 3 times with water, and we added 15 milliliter of liquid MSR medium with our treatment is fungicide or controls to the hyphal compartment. As a fungicide, we tried the effect of iprodion, pyraclostrobin, etridiazole, and fludioxonil, all in the field concentration with 2 controls, water and acetone. So we took sample in the time 0 and every 12 hours until 72 hours. And then we analyzed the samples by spectrometry device ICP OES. So based on the results, the transport of phosphorus from the interradices to the plant in the presence of pyraclostrobin and fludioxonil, it was less than the acetone control. In another experiment, we verified the effect of pyraclostrobin and iprodion on phosphorus uptake by extra-radical mycelium of interradices in the pot culture conditions. In this experiment, we use 1 liter pots and we use 10 centimeter pipe tubes that we made 1 centimeter holes around the tube, 31 holes, and then we cover the tube with the 41 micrometer membrane that just lets the mycelium and hypha pass through the tube, not the root of plants. So then we fill the pot with the mixed sand and we put a layer of interradices inoculum, and we placed the empty tube inside that was covered in the bottom with the plastic Petri plates. So we placed the seeds of Medicago, and after almost 1 week, 10 days, we had plants of Medicago that the roots were in contact with the inoculum of AMF. After 2 months, when they got colonized with the AMF, we filled the tubes with the mixed sand. We had 3 controls: non-mycorrhiza, water, and acetone, and 2 fungicides, pyraclostrobin and iprodion. So we added them to the tubes with 50 microliter water, and then after 1 month, when the mycelium penetrated through the holes inside the tubes, we collected them for analyzing. We also collected the roots in the pot and mycelium in the tubes to have a gene expression experiment, and it's ongoing. So based on the results, Enterodices in the contact with iprodion made 3 times more mycelium compared to acetone. And also treatment pyraclostrobin showed less arbuscule abundance, and iprodion had more vesicle abundance. But still, we are analyzing them, and it's ongoing. And this is my supervisory committee from University of Louvain, ENAC company, and Hydreka company, and I want to thank your attention.
Thank you, Marjan. Okay, so then we can pass to Kara.
Okay, 1 moment. Okay, you can see my slide.
Yes.
Okay. So hello everyone, I'm Kara Meyer, and I'm going to talk to you about how soil microbial community fragmentation reveals indirect effects of fungicide exposure mediated by biotic interactions between microorganisms. So yeah, as I said, the first half of my PhD was at INRAE in France with the supervision of Laurent Philippot, and the second half at Syngenta Stein, supervised by Claudio Scarpanti. So we know that, well, fungicides are an essential part of modern agriculture used to protect crop yield and quality, but they can have effects on off-target organisms, including microorganisms. They can cause increases or decreases in microbial diversity, abundance, and functions. But it's important to keep in mind that these are net effects, which include both direct and indirect effects. So what do I mean by indirect effects? Well, we know that microbial taxa interact in a microbial community. So when we apply a pesticide, we will expect not only direct effects on taxa that are sensitive to that pesticide, both non-target and target taxa, but also indirect effects through, so on taxa that are not themselves sensitive to the pesticide, but are interacting with sensitive taxa. But what we want to know is how widespread are these indirect effects in soil microbial communities? So to dig into this question, we fragmented a microbial community in order to modify the biotic interactions between taxa. We then compared the fungicide effect across our different fractions. So for example, here, if you look at the top row in our unfragmented community, we see that when we apply our fungicide, there's a decrease in abundance for both this orange fungus and the blue bacterium. And then if we look at our fragmented communities, we see in one fraction that there's still a decrease in abundance on this orange fungus, but this blue bacterium, there's no effect. So by comparing these responses to the fungicide across our different fractions, we can surmise that this orange fungus may be directly affected, whereas the blue bacterium is probably indirectly affected by the fungicide. So as a model fungicide, we use timexazole. This is used worldwide for soil-borne fungal plant pathogens. It's used as a soil or seed treatment. It's a DNA RNA synthesis inhibitor, and it's relatively bioavailable as it has it's quite mobile in the soil and moderately persistent. So here's a more detailed view of my experimental design. So we took our original soil and we made a soil suspension, and then we filtered it in order to fragment the microbial community. So we end up with 5 different communities, including our unfiltered or unfragmented community. And then we treated those 5 different communities with 4 different hymexazole treatments. So including our no hymexazole control, and then 1 time the recommended field application rate, 10 times that rate, and 100 times. So we then, with our 20 treatments, we had 10 replicates of each, so a total of 200 microcosms. So these different treatments were inoculated into sterile soil microcosms, which had been prepared with the same soil which was used to make the soil suspension, but it had been sterilized by gamma irradiation. We then incubated these microcosms for 40 days to allow recolonization of the sterile soil by the different communities. And we reapplied the same dose of hymexazole halfway through to all microcosms. So at the end, we then harvested our microcosms and sequenced 16S and ITS in order to look at the bacterial and fungal communities. So to look at the results, but also I should mention that I'm going to just talk about the bacterial results because basically this approach seems to work better for bacteria than for eukaryotes. We saw a lot of variability even within treatments for the fungal data. So first of all, we see that our fragmentation approach worked. If we look at just the controls for each of our different communities, we see that there's a change, like fragmentation altered the microbial community. And also we saw an effect on alpha diversity, beta diversity. We also see that there are dose effects. So for example, here we see that as dose increased, there's an increase in the relative abundance of firmicutes and a decrease in the relative abundance of alpha proteobacteria, for example. But importantly, we see that the dose effect differed across fractions. So it's not the same in all of these communities. We can also see that hymexazole and fragmentation affected certain functional groups. So we've been talking about ammonia oxidizing bacteria. This is looking at qPCR data, so looking at the MOA for ammonia oxidizing bacteria. And we can see that the copy number decreases generally with hymexazole concentration. But again, it's not the same in all fractions. The response differs across our different fractions. So this suggests that indirect effects could contribute to changes in ammonia oxidizing bacterial abundance. So that soil functions such as nitrogen cycling can be impacted by indirect effects. Okay, so in order to find and identify our hymexazole affected OTUs, we performed a differential abundance analysis where we compare the relative abundance of OTUs to the control for each fraction. So in the center here, we see the phylogeny of the OTUs which were affected. So this is the 473 OTUs which were affected by hymexazole in at least one treatment. This ring here shows us the taxonomic identification of the OTU. And then each ring shows us the change in relative abundance of each OTU at each dose across the different fractions compared to the control for that fraction. So it's a lot of information, but generally what we can see is that there are taxa-specific responses. So again, we see that firmicutes tend to increase in abundance with higher hymexazole concentrations. Alpha proteobacteria, again, tend to decrease in abundance with increasing hymexazole concentrations. But we also see that very few OTUs respond consistently across fractions and doses. So we come back to our question, how widespread are indirect effects in the microbial community? So this chart shows us the number of affected OTUs in each fraction at each dose. And the dark gray are showing the number of OTUs which had a different response across fractions. So those are our indirectly affected OTUs, whereas the light gray is showing us the number of OTUs which had the same effect across all fractions. So as you can see, most bacterial OTUs had a different effect across different fractions. So this leads us to conclude that indirect effects are the predominant type of effect in the microbial community. Okay, so then these white numbers now are showing the percentage of affected OTUs which had a different response across fractions. So we can see that although the indirect effects in all treatments are still dominant, still the most common type of effect, as dose increases, we see that there are fewer indirect effects, so that indirect effect actually decreases at our 100x dose. So this kind of makes sense that with a higher dose, you may see more direct effects. But it's also important to point out that we may still be underestimating indirect effects because, of course, with our only 3 filters, we didn't alter all biotic interactions. Okay, so then let's look at the potential direct effects. And again, I say potential because it may just be that we can't see that they're indirect. But so we see, again, there are more direct effects as dose increases. And interestingly, more of these were increases in relative abundance as dose increased. So this may suggest that hymexazole could be used as a nutrient source for certain bacteria, leading them to thrive and increase in relative abundance. Okay, so in order to investigate the interactions that could underlie these indirect effects, we performed a network analysis. Instead of showing you the network, I'm just going to show you the number of edges in our different networks at the different doses. So these are the edges which are related to hymexazole. And we see an increase in hymexazole in related edges at 100x. So here. What's interesting is that although the edges between fungi and bacteria and fungi and other fungi increase at 100x, they like double. The bacteria to bacterial edges actually quadruple. So this is suggesting that although this is a fungicide, it has a large effect on bacterial interactions. So in conclusion, we found that fragmentation allowed us to disentangle direct and indirect effects of hymexazole exposure. We saw that 13% of the dominant bacterial OTUs were affected by an agronomic dose, that is the 1x dose of hymexazole in the unfragmented community. And 85% of those effects were indirect effects. So indirect effects decreased at higher doses, but they were still predominant. And our network analysis provided some insight into the interactions which could underlie these indirect effects. Thanks. Okay.
Thank you, Kara. And so we'll move to the final presentation by Kuni Yang.
Okay. All right. Okay. Hello, everyone. My name is Kun Yang. In our project, we want to develop computational tools to prioritize pesticides and their transformation products for soil microbial ecotoxicity testing. Yeah, so we focus on a very important process in soil, which is nitrification. It is critical to global nitrogen cycle, and it is controlled by ammonia oxidizing microorganisms. Once pesticides are applied, both pesticides and their transformation products can affect the activity of AOM. And it has been reported that some transformation products can have even higher toxicity than their parent pesticides. So there are two goals in our project. The first, we want to correctly predict transformation products of pesticides. And then we want to predict the toxicity of pesticides and transformation products on the nitrification activity of ammonia oxidizing microbes. So in the end, we will have a comprehensive toxicity evaluation for the whole transformation pathways of pesticides in soil rather than just parent pesticide. So first, in order to predict for the transformation products, we need to let the model learn from existing biotransformation pathways. So fortunately, we have EnvyPath. It is a database of biotransformation pathways, and there are two major data packages in EnvyPath. The first is EVOC-BBD. It contains biotransformation pathways observed in pure or enrichment cultures, while the other one, EVOC-Soil, has biotransformation pathways observed in soil. And the model we use is a hybrid model. It combines chemical knowledge and machine learning models. The chemical knowledge in our case is a set of biotransformation rules. Each rule represents a type of reactions. So for example, here we have a biotransformation rule that oxidize hydroxyl group to ketone or aldehyde. So then the machine learning models will decide what biotransformation rules to trigger, so then we can get transformation products. So as you can see, the biotransformation rules are very important for our prediction. Before in EnvyPath, we have more than 200 biotransformation rules that were manually designed by experts. But we recognized two potential problems of the manual rules. The first is when we add new reactions into our database, it's very difficult to update those existing manual rules to cover new reactions. And then the second problem is some of the rules are very general, meaning that they can be triggered on many compounds and cause high false positive rate. While on the other hand, some rules can be very specific and they only work for one or two compounds. So in the end, we might miss some important transformation products. Both of these two problems can hurt the prediction performance. And to overcome these problems, we develop a tool that can automatically extract rules from biotransformation reactions. This tool is called EnvyRule. So first, we send all the biotransformation reactions into reaction clusterer. Then the reactions will be clustered into different reaction groups based on the reaction fingerprints. Then for each group of reactions, the rule will be automatically extracted. And later on, if we have new reactions, we can send them into reaction adder, and then the reaction groups will be updated. And also their corresponding rules will be updated. So in the end, we can send the automatic rules back to NV paths for the prediction of transmission products. We compared the prediction performance of models with manual rules and automatic rules. And based on the precision recall curve of the model, we can see that the model with automatic rules has greater area under the curve score than the model trained with manual rules, suggesting that automatic rules improve the prediction performance of transmission products. Then for the second task, prediction of toxicity towards nitrification, normally it is done by in vitro tests that people apply chemicals with different concentrations on ammonia oxidizing microorganisms to see whether the nitrification activity of AOM can be affected. And as you can see, the test normally runs more than a month. So we propose to use model prediction. First, the model will convert atoms and bonds into numeric representation. Together with the adjacency matrix of the atoms, they are sent into the model as the input. Then there will be information exchange between atoms during the convolution layers. And we also have a multi-head attention component that can recognize important structures from the molecule that are critical for the prediction. And in the end, we can either predict for a specific property of the molecule, or we can classify whether this molecule is toxic or non-toxic. And this model is called graph neural networks. The challenge of our model prediction is we could only find about 300 compounds that has the information of toxicity towards nitrification. And it is a very small number of data sets to train deep learning models, and which may cause very bad prediction performance. Our strategy is we first train the model with a data set that is relevant to toxic, but has much larger data size. Nitrification is controlled by ammonia monooxygenase, AMO, and it is a membrane-bound enzyme. And it is known that the compounds with higher log P values are more likely to affect the functionalities of membrane, including the membrane-bound enzyme. And people have already used log P values in QSER models to predict nitrification inhibition. It has a very decent performance, which means that log P and toxicity towards nitrification are related. So we pre-trained our model with a log P dataset that has about 100,000 compounds, and we managed to get a very good R-square score, which is 0.95. Then we fine-tune the model with toxicity towards nitrification dataset that has about 300 compounds. And in the end, our model achieved 85% accuracy, which is very good. So yeah, that's how we improve the model training through mechanistic understanding. Although we already have a model with good prediction performance, we are not satisfied yet. We want to know what chemical structures are important for the prediction. So with the help of our attention mechanism, we can identify important structures and less important structures. Here I take log P prediction as an example. So the dark green parts are the important structures automatically recognized by the model, while the dark purple parts are not so important structures, and they make a lot of sense. However, we have one more question, is we don't know, or we are not sure whether the important structures make positive or negative contributions to the prediction. So here is an example. In the first row, those two important structures, the aromatic chloride and aliphatic sulfide, they increase log P. So they make positive contributions to log P. Well, in the second row, this aromatic amine and this nitrogen group, they lower down log P, so they make negative contributions. But they are all, these four structures, they are all important. So we need to use Shapley values to determine the contributions of the important structures. And Shapley values can calculate the contributions of a substructure conditioned on the whole molecule. So for the same substructure, it will have different contributions in different molecules, and that makes sense. For example, for this molecule, we first use attention mechanisms to identify important structures for the log P, and then we use Shapley then we calculate their Shapley values. And for these two nitrogen groups, they make negative contributions to log P, while for this carbon, oxygen, nitrogen chain, it increased log P. So then we can use attention mechanisms to identify important structures and use Shapley values to find the contributions of the important structures. We combine those strategies and apply them on our toxicity dataset. So then we could find the structurals that are responsible for the toxicity towards nitrification. So they make positive contributions to the toxicity. Here are some examples. Some of the structurals, they have nitrogen-carbon double bond, and some have sulfide nitrogen. We have a total of 24 structurals identified from our database. And among the 24 structurals, we are particularly interested in a specific type. Knowing that ammonia monooxygenase is a membrane bound enzyme, and also compounds with higher log P values are more likely to affect membrane. So we might assume that the structurals that increase log P should also increase the toxicity. However, by comparing the structurals of log P and structurals of toxicity, we notice that that's not the case. We also found structurals that can decrease log P, but increase the toxicity. And by doing more research, we realized those compounds, the compounds with those structurals, they might serve as the substrates of ammonia monooxygenase. So that is a very different inhibition mechanism. So by comparing the structurals from different data sets and different models, we can expand our mechanism understanding. So here comes the conclusion. In the first task, prediction of transmission products, the MV rule can automatically extract rules from reactions, and the prediction performance of transmission products can be improved by using automatic rules. And then in the second task, prediction of toxicity towards nitrification, we applied transfer learning with log P dataset to improve the prediction of toxicity. And we also applied attention mechanism and Shapley values to identify important structures and also their contributions to model prediction. And I just showed that the analysis of structurals can potentially help for a more comprehensive mechanism understanding. And that's it. Thank you very much.
Excellent. Thank you, Kuni Yang. So I would like to thank everyone for presenting, and maybe now we can have a bit of a discussion on the stuff that the fellows have presented. And I have some backup slides if we need to discuss a bit further, just to let you know that we are actually working on the establishment of on the use of SSDs and principal response curves into an ecotoxicological context using amplicon sequencing data and data derived from our in vitro tests in order to establish to be able to use amplicon sequencing data in for the ecotoxicological assessment of effects of pesticides on the soil microbiota and get quantifiable results. And also, just to let you know that some of the tests that Eleftheria has presented, and of course, Marjan's tools are already miniaturized, but some of the tests that Eleftheria has presented for our ammonia oxidizers, we have been working in collaboration with ECL and other groups outside ARISTO to develop them into high throughput systems testing, so in 96 well plates, in order to provide a fast track assessment of potential effects on the ammonia oxidizers. So, yes, so Agnes.
Yeah, so first, thank you very much for these very, very